Hi, my name is Mikael Rydén. I'm an endocrinologist. I'm going to, in the next 30 minutes or so, present a bit of data that we've generated over the last couple of decades and try to summarize why it's important to study white adipose tissue. So the outline I will have in my presentation will be the following. So I'll talk a bit about the role of the adipocyte as a metabolically active organ. And then the difference between hypertrophic and hyperplastic obesity and why that's important to understand. And then the effect of current and future obesity treatments on adipocytes. So I did my PhD in molecular neurobiology. So when I moved into the fat field, I felt kind of strange because I was moving from an organ that everybody understands is really important, the brain, and into the fat tissue, which is basically a tissue that most people want to have as little as possible of. But it's an extremely important tissue for metabolism and for metabolic diseases. So first of all, if we put adipose tissue in perspective, so a few things we need to take in consideration is the fact that there are many different adipose tissue depots. So you have the abdominal depot, which can be subdivided into the subcutaneous and the visceral adipose tissue. There are many different parts in the visceral adipose tissue. We often study the omental adipose tissue because that's basically the curtain in front of the intestines, and it's easy to get biopsies from them, from that part during surgery. And when it comes to subcutaneous adipose tissue, we mostly focus on the tissue that is in the periumbilical area, and that's because that's metabolically quite active. So if you compare that to gluteal or femoral tissue here, the activity in these adipose tissue depots is much lower, and there's actually a protective effect of the function in these tissues because they're so metabolically inactive. So what we see is that these tissues are the ones that are really positively correlating with metabolic disease. That's why we study these depots. So an obvious caveat in humans is that visceral adipose tissue cannot be studied longitudinally. You cannot get access to visceral adipose tissue in longitudinal studies. You can only get that from the subcutaneous depot. And that's very easy, so you can get several grams by subcutaneous biopsies. What has also become clearer in the last 2 decades is that there are very different ways in which adipose tissue can expand. So there's the good way of expanding your adipose tissue, which is depicted here up, where you see that despite the fact that you expand the tissue quite considerably, you don't have an increase in insulin resistance, you have an increase in adipocyte proliferation, so more fat cells produced. You don't have any changes in lipid turnover or inflammation, and no fibrosis or changes in vascularization. This phenotype, where you have many small fat cells, is called hypoplasia, and I will come back to that later, why that is so important. That can be compared with a metabolically pernicious way of expanding your adipose tissue, which is shown here, where you have an increase in insulin resistance. You don't have an increase in adipocyte proliferation, so you actually have an increase only in the adipocyte size. That is also linked to increased lipolysis, inflammation, fibrosis, and a reduction in vascularization. So we usually define this as a hypertrophic adipose tissue. And the problem is that we all are born with different ways of expanding our adipose tissue. We don't know why, but some people have a tendency to increase adipose tissue via hyperplastic mechanisms, and they are protected against diabetes and dyslipidemia with weight gain, while others tend to grow their fat cell size, hypertrophy, and then they tend to increase the risk for metabolic diseases such as type 2 diabetes and dyslipidemia. So an important thing to understand about white adipose tissue is the pathophysiology of free fatty acids or non-esterified fatty acids. So from the visceral adipose tissue, the fatty acids, the non-esterified fatty acids are transported through the big route here, the portal vein, directly into the liver. And the fatty acids are then used in order to make more glucose or gluconeogenesis. That results in increasing insulin production from the pancreas, and you also use these fatty acids to produce VLDL triglycerides. Conversely, in the subcutaneous adipose tissue, when fatty acids are released into the peripheral veins, the fatty acids are preferentially used by the muscle, the skeletal muscle, and the skeletal muscle actually prefers fatty acids to glucose, and that leads to a decrease in insulin action and a decrease in glucose uptake. So you tend to keep your glucose in the blood instead. And another thing that we know from fatty acids, at least in animal experiments, is that free fatty acids have a detrimental effect on beta cell function, so they decrease insulin secretion. So this means that if you release fatty acids, and that leads to a constant elevated level of these free fatty acids in the blood, then that promotes an insulin-resistant state. Another very important thing to understand about fat is that adipose tissue is the best place to keep lipids. So lipids are stored primarily in the subcutaneous adipose tissue, which has a large capacity to expand, as we all know. And when storage capacity is attenuated, this leads to ectopic lipid deposition. So if we have here our subcutaneous adipose tissue, and it cannot retain the lipids that we feed it with, then they will be ectopically deposited into the liver, leading to non-alcoholic fatty liver disease, into the pancreas, leading to lipotoxicity, as I said, skeletal muscle, reducing insulin sensitivity. Lipids can also be stored in the vessels, which leads to atherosclerosis in the long run. And also, you can deposit the lipids in the visceral adipose tissue. So in our, the current view of how white adipose tissue promotes metabolic disease is that the subcutaneous adipose tissue is the real culprit. So when we all talk about visceral adipose tissue as the bad guy, that's just because we don't have sufficient amounts of subcutaneous adipose tissue or sufficiently good function to store our lipids, and then they are funneled out into the visceral adipose tissue. So the visceral adipose tissue is basically an ectopic fat depot, and we all know that ectopic fat can be very good to eat. This is foie gras, which is basically NAFLD in ducks, but we certainly don't want to have that in our bodies. So what have we shown regarding the association between adipocyte lipolysis, so lipid turnover basically, and cardiometabolic disorders? So we showed a couple of years ago that in over 1,000 individuals, differences in lipid uptake in the subcutaneous adipose tissue explain 20% of the variation in the metabolic syndrome score. So no matter what you corrected for, all aspects, the differences in adipose tissue function explain 1/5 of the variation in your metabolic risk. Similarly, in 1,000 subjects, we also showed that the lipolysis measured explained 14% of the variation in the circulating HDL cholesterol and triglycerides. So again, fat tissue contributing to some extent to important cardiometabolic risk factors. So our take on that was that in large cross-sectional analysis, dysfunctional lipid handling in subcutaneous adipose tissue associates with cardiometabolic risk factors. Now the problem with this is that this is only, these are only correlations, and you can't say anything about causation. So another thing we've done then in order to understand the causality of lipid turnover and disturbances in lipid turnover is to do carbon-14 dating. So when you have a fat cell in the lean state, you have a balance between the lipid uptake, lipid storage, and the lipid removal. So you take up as much as you release, and that gives you a specific size of your fat cell. Now, the balance between lipid uptake and lipid removal can actually be reflected if you take out the lipids and measure their age by carbon-14 dating. So that you can actually measure the triglyceride age, which is approximately 1.6 years in a fat cell. You can also, based on this parameter, calculate the amount of lipid storage that has been delivered into the fat over per year. So the kilograms stored fat per year. That's the K in. And then you can also calculate the lipid removal based on this, which is basically the inverse of the triglyceride age. And that's the K out. And normally there's a balance between K in and K out. In the overweight state, what we were able to show was that the uptake of the lipid storage wasn't altered in the overweight state. But the lipid removal was considerably and significantly lowered. So that led to an increase in triglyceride age. And similarly, in obese subjects, we could show that in these subjects, there was not only a disturbance in lipid removal, but also an increase in lipid uptake. So that gave an even further increase in triglyceride age. So we can actually measure these parameters, at least some of them, in our clinical cohorts, and that's lipid removal, which is basically the same thing as lipolysis, triglyceride hydrolysis. And we've shown in several papers that there is an inverse relationship between the stimulated lipolysis and lipid age. So the more you can activate your lipolysis here in the subcutaneous adipose tissue, the lower the age, which would fit with this model. If you're good at removing your fat, you get a lower age in your triglycerides. And we've shown the same thing in visceral adipose tissue. Now, all this is good, but it's just cross-section analysis. So what about causality? Can disturbances in adipocyte lipolysis promote weight gain and type 2 diabetes? So we started this project a couple of years ago trying to see in prospective analysis whether disturbances in adipose tissue function could actually predict weight gain and type 2 diabetes development. So in order to address this, we recruited women that had been followed for at least 13 years in our laboratory. And we looked at the incidence of weight gain and type 2 diabetes and prediabetes at the follow-up, so after an average 13 years. And here you can see them at baseline. So you see that we could select 18 women that in the end gained weight, and we had 36 that were stable, and at the baseline examination there was no difference in body weight between the two groups. However, after 13 years of follow-up, actually 13.4 years of follow-up, you see that the weight gainers had on average increased their body weight by 13 kilograms, while the weight-stable subjects had basically not changed their weight at all. Now, an increase in 13 kilograms over 13 years may sound extreme, but it actually, if you calculate the number of extra kilocalories that you need per day in order to gain that much weight, that equates to approximately 10 to 25 kilocalories per day. So it's basically a sugar cube per day extra. So you could never find these differences by looking at what they ate or how they moved or things like that. Okay, so we have these two groups of women followed for a long time. Some of these women went up and some were completely stable in weight. And then we tried to see were there any differences at baseline that could explain why some were weight gainers and some were weight stable. So this is a very busy table, but it basically shows that there were no differences in anthropometric parameters. They were equally chubby at baseline. They had the same age. There were no metabolic differences. They had the same respiratory quotient, the same HOMA, total cholesterol, leptin levels were exactly the same, and there were no differences in lifestyle. So for instance, we know that pregnancy and giving birth to many kids is a risk factor to gain weight, but there were no differences in the number of children that these women gave birth to, or menstruation, or anything like that. So the only thing that we actually found was different was that the weight-stable subjects had a much higher stimulated lipolysis, so they could actually move their fat much faster than the weight gainers could. And this difference is quite extreme. It's a 30 to 40% difference in lipolysis. So the conclusion here was that weight gainers had a high basal but low hormone-stimulated lipolysis at baseline. And this was the only thing that could be used in prospective analysis. And that's shown here, where you see the delta BMI and the stimulated lipolysis on the x-axis. And you see that the more you can stimulate your lipolysis, the more you can move that, the smaller the difference in BMI over time. So the question was whether there was any heredity, differences in heredity when it came to weight gain, type 2 diabetes, things like that. No difference. We asked that as well. So the family history was exactly the same. There was nothing that could identify these ladies at baseline. This was the only thing. Now, so we were pretty happy when we found this. So we submitted it to a big journal on the East Coast of the States, and hope to. And we got one question, and that was, of course, the usual thing: can you show this in a replication set? Can you validate this in another set of individuals? So I spent most of the Christmas of 2017 looking in our data sets, and I actually found 25 women that we'd followed for 10 years, and where we could see exactly the same relationship. These ladies had nothing to do with the other ones. And we could also see that the lipid age was much older in the ones that gained weight. Now, what are the risks if you had a low basal lipolysis at baseline for different metabolic risk factors? So if you look here, these are the odds ratios. So for weight gain, there was a 6-time higher risk for weight gain if you had low lipolysis at baseline, around 4 times higher risk for type 2 diabetes, and an increase in insulin resistance of almost 9 times. It was as a 9-fold increase in the risk of that. So we never published that in that journal. We published it in Cell Metabolism instead. But basically, what we showed in this paper was that in the lean state, there is a balance between lipid uptake and lipid removal. And you have a good way of activating your fat. If you have a low lipid removal at baseline, this leads to an increased risk to develop overweight and obesity, and thereby type 2 diabetes. And the best way of comparing this would be with an engine. So on the left, the healthy way of showing your lipolysis, the low basal and high stimulated, well, that would be an engine where you have a low idle RPM, but with a possibility to, when you push the gas, you can really increase your lipolysis. On the other hand, the other extreme then, when you have a high basal but low stimulated, would be an engine where you have a very high idle RPM. And when you push the gas, you really can't increase the lipolysis very much. And the equivalent in the car world would be a Ferrari for the left and a Fiat Cinquecento for the right. But remember that this doesn't say that one thing is good and the other one is bad, because this is all based on the fact that this is how our stores look like today, our supermarkets. We have 32 different kinds of mayonnaise. And of course, under these circumstances, it's really bad to be, look like a Fiat Cinquecento. But on the other hand, it wasn't a long time ago that stores in Europe looked like this. And in that case, it might actually be better to be the Fiat Cinquecento, because then you are much better at retaining your lipids and energy, basically. So you can withstand starvation in a much better way. So I would say a few words about the difference between hypertrophic and hyperplastic obesity, which I started with. And when it comes to the increase in body fat mass and fat cell volume, there is actually not a linear relationship. You have this curve, the curved linear relationship, where you can see that this is admittedly based on cross-sectional data, but you could infer from this that perhaps when you increase slightly in weight, you have an increase primarily in fat cell volume. However, if you increase in weight from an already obese state and you become super obese, you don't have an increase in fat cell volume. You have to increase your fat cell number. But you can also see that there are a number of individuals. This is the mean curve that you see here. But you see for each body fat mass, there are subjects that have very, very large fat cells and subjects that have very, very small fat cells, even though they have exactly the same body fat mass. In fact, there are some obese people here that have smaller fat cells than very, very thin people. And what does that mean? I think I skipped this. So this means, and we've shown this and many others have shown this, that if you have a hypertrophic adipose tissue, you actually have a very much higher risk of developing type 2 diabetes. And subjects with hypertrophy in their fat cells also have a much higher risk of being have a hereditary history of type 2 diabetes in their family, while hyperplastic adipose tissue seems to be protected. So what about differences then in adipose tissue cellularity if you have it in the subcutaneous or in the visceral adipose tissue? So regional differences in adipose tissue cellularity may be of importance. So if you look at the relationship between fat cell size in the subcutaneous adipose tissue, you see that there's a very close relationship to insulin resistance, while big fat cells in the visceral adipose tissue correlates primarily with dyslipidemia. And what about fat cell number? So if you have many fat cells in the subcutaneous adipose tissue, that's linked to a beneficial metabolic phenotype. In contrast, if you have a large number of fat cells in your visceral adipose tissue, that has no relationship to clinical phenotype. So our conclusion from that is that the visceral adipose tissue is an ectopic fat depot, and there's no benefit of having many fat cells in there. So what about the effect of current and future obesity treatments on adipocytes? So I'll tell you a few things about what happens with adipocyte number and weight loss. So these are subjects that were followed before and 2 years after bariatric surgery. They lost quite a lot of fat, and you see that the fat cell volume decreased in the ones that lost weight after 2 years. However, if you look at fat cell number, there was no difference at all. So fat cell number seems to be constant after weight loss, at least after 2 years. What about changes in weight if you gain weight? So here we had a group of subjects that were weight stable or weight gainers, and they were followed for 10 years. And you see that the weight stable ladies here, they were completely weight stable, no change in BMI, while the weight gain is really gained from 23 up to almost 30 in BMI after 10 years. And this was not because they were training and getting more muscle, they really got more body fat mass. So no difference here in the weight stable ladies, but the weight gainers got a huge increase in fat mass. Fat cell volume, what happened with that? Well, nothing in the weight stable subjects, but the weight gainers got an increase in fat cell volume. And finally, what about adipocyte number? No difference in the weight stable subjects, but a significant increase in the weight gainers. So summarizing this was also corroborated by other studies, but what we conclude from this is that fat cell number can increase but not decrease. Once they've been formed, a new set point is set. So the analogy would be these plastic straps that you put around your cables or your garbage, and which you can only pull them in one direction. You can never go back. So I would say that that's the same thing with fat cells. You can only increase your number of fat cells. You can never decrease them. And that's one of the reasons why it's so difficult to lose weight. Because once you've been overweight or obese, it's extremely hard to retain a lower body weight. And that's been shown by us and others that the fat cells that you have after you've been obese and then lose your weight are very hungry. They want to become big again. And that's one of the reasons we think contribute to the fact that we have this type of yo-yo banding in humans, where you lose weight. Everybody can lose weight. You just put them on a Robinson Island and everybody will lose weight. But most of them will regain. So my final slides here is just about how the adipose tissue phenotype is altered by long-term weight loss. And this is unpublished data. We mapped the insulin-induced transcriptome in human adipose tissue before and after pronounced weight loss and compared to never obese subjects, because we wanted to understand how the adipose tissue is changing. And we had, so basically we had three groups. We had the never obese here, NOs, we have the obese that are here, and then we have the subjects that were obese but lost body weight after 2 years, and we call them post-obese because all of them had a BMI below 30. And we did biopsies, subcutaneous biopsies, before and after a 2-hour hyperinsulinemic euglycemic clamp. So we got the biopsies before and then 2 hours after, when they had been bathing in a lot of insulin in vivo. And then we looked at the insulin response in these. And you see here the NOs, the never obese subjects, they respond quite extensively to the insulin. So they start here. This is the fasting and this is the hyperinsulinemic condition. Here we have the obese. They are very similar in the fasting state, but clearly different in the hyperinsulinemic state. And here we have the post-obese. So you see that the post-obese, they actually move up. They do not only normalize, they actually super normalize, it seems. And that can be shown in another way, which is done here. So here we look at the genes that respond to insulin in the three groups. So here you have approximately 280 genes that respond, 250 up and 31 down. Here in the obese you have around 100 genes that respond, most of them up. And here in the post-obese you actually have almost 490 genes responding, so quite a lot of genes responding in the post-obese state. So one can note a few things here. First of all, the response is much lower in obese subjects than in post-obese subjects and in the NOs. But the post-obese actually respond more than the never obese subjects, despite the fact they have exactly the same BMI. And when you look at the genes, you can identify the genes that are the core set that are changed and responding in all three conditions. This is the core set of genes. And then you have the yellow ones here, which are only responding based on weight. So they are here in the never obese, they are completely lost in the obese state, but they come back in the post-obese state. And then you have some genes that are only present in the non-obese, the never obese, and then you have in the green there, you have the ones that respond only in the post-obese. So I think that this really shows you that weight loss changes completely the architecture of adipose tissue. Once you lose weight, you actually get a supernormal response to insulin. And my final slide is just saying a few things on how should we target adipose tissue in the clinic. So could we increase fat cell number in the subcutaneous depot? This has already been tested by the glitazones. And they work mainly by increasing fat cell number and normalizing fat cell function. However, they have side effects outside of the adipose tissue, which makes them less good to use in type 2 diabetes treatment. Could we improve insulin sensitivity based on some of the data that we've generated? Could we identify specific pathways that could be improved? There are also fat cell specific biguanides, as metformin analogs that we've tested, and others as well. Could one attenuate basal lipolysis, reduce local inflammation? I haven't shown much about that. But also the final thing is to alter the endocrine function of adipose tissue. I haven't talked so much about leptin and adiponectin, but it's possible that tampering with the adipose tissue endocrine function could also be of importance. So to summarize, what I've tried to say is that cross-sectional studies demonstrate an association between fat cell size, dysfunction, and cardiometabolic complications. Long-term prospective studies suggest a causal role for altered subcutaneous lipolysis and weight gain in type 2 diabetes development. Fat cell hypertrophy is associated with altered lipid turnover and local inflammation. Weight loss, irrespective of method, decreases fat cell size but not number. Transcriptomic analysis of the insulin-induced response in human adipose tissue reveals that insulin resistance is normalized upon weight loss even beyond the normal level, and that future weight loss approaches targeting adipose tissue may need to involve multiple pathways. And I just want to acknowledge all the work and the collaborators and the funding bodies that we've had. Thanks. 12:45. So the question was regarding polycystic ovarian syndrome. So it's very well known that PCOS is a polygenic syndrome, which is very common. Up to 10% of all women have that in Sweden. We know that, for instance, the PCOS phenotype deteriorates a lot with obesity. And we've looked at what happens with the adipose tissue in PCOS, and we can identify specific regulators of lipolysis that are altered in the polycystic ovarian the PCOS ladies compared to non-PCOS women. And these are parts of the PKA pathway, PRK beta 2 and PRK R1 alpha. So we know that there are I don't know if that's the entire explanation, but we definitely see specific differences in the adipose tissue of polycystic ovarian syndrome women. All right, good luck with your lives.